So we are going to go with the second short talk, which will be from Ève-Marie Frigon, who is a PhD candidate in biomedical sciences in the anatomy department of the University of Quebec in Trois-Rivieres. And today she's going to present her work on histology, MRI and T2 star registration quality of ex vivo human brain blocks fixed with solutions used in gross anatomy laboratories. Yeah, you can go ahead. Thank you.
Hi, thank you for the introduction. My name is Ève-Marie, and I will be presenting our work on histology, MRI, and T2 star registration quality of ex vivo human brain blocks that were fixed with solutions that are used in gross anatomy labs. Whoops. Sorry. So as you all know, MRI is the most common neuroimaging modality in neuroscientific research. It is widely used for its capacity to show anatomical changes in a non-invasive way. The images that are obtained with this modality are generally gross anatomy 3D images, and it helps the user to be oriented in the brain. However, it doesn't provide cellular resolution, and to have such resolution, microscopy of histology sections is still the gold standard to show cellular changes happening in normal aging or in neurodegenerative brains. And so histology also provides a higher resolution, but results in more distortions. And it's also more difficult for the users to locate or orient structures since it refers to very thin 2D sections. And to have a best of both worlds, registration between MRI and histology also validates MRI findings by showing the cellular changes found in the MRI, by also increasing the ease of colocalization. And since histology is still a gold standard and that MRI findings needs to be validated, neuroscientists are in need of histology tissue that is provided in vivo by biopsies. Well, since they are very invasive and provide very small tissue samples, it is also almost impossible to use them in neuroscientific research. So therefore, people use postmodern human brains, but the tissue is nowadays obtained through brain bank that provide very small and few tissue blocks in comparison to what we could obtain from gross anatomy labs, which is a higher amount of full brains. However, these brains are fixed through whole body perfusion with chemical solutions that are used for body dissection. These different chemicals may change the cellular morphology by interacting with different proteins, since the solutions used in gross anatomy labs differ in their chemical composition than the classic neutral buffered formalin that is used in brain banks. For modern neuroscientific research that we know, we ignore if the cellular morphology or the tissue quality or antigenicity of the main brain cell populations could be preserved in these kind of brains. A previous study of our team showed that the neutral buffered formalin that we'll call NBF, that is used in brain banks, and two other fixatives that is used in our gross anatomy laboratories, such as saturated salt solutions that we'll call SSS, or an alcohol formaldehyde solution, or AFS, preserved antigeny of the four main cell populations in mice brains. And so our goal is now to compare the histology, MRI, and registration quality of the human brain samples that were fixed in our anatomy laboratory in comparison with the ones fixed with NBF in brain banks. So we expected a better histology and MRI quality using NBF fixed brains and a more difficult registration using SSF solution due to the macroscopic texture that was very different between the brains and that could enhance tears in the tissue for the salt blocks. So overall, our goal is really to assess the potential use of brains from anatomy labs to increase the amount and the quality of brain tissue available for neuroscientists, and to have a proof of concept of a good MRI histology registration pipeline with those kind of brains. And to do so, we use the convenient samples of 42 brains coming from first the Douglas Bell brain bank for the NBF cases, which were fixed by immersion of the brain, while the other cases were obtained in our anatomy lab and were fixed by whole body perfusion. This is the procedure, like the common procedure in anatomy labs, in which the solution is injected through the carotid artery, as you can see here, with a pump. And in our case, it's injected with a SSS, a salt-saturated solution, or the alcohol formaldehyde solution for a full body involvement for dissection. And for histology assessments, the 42 brains were sectioned at 40 microns and processed with immunohistochemistry of the 4 main cell populations, such as neuron for neurons, GFAP for astrocytes, IDA1 for microglia, and PLP for oligodendrocytic myelin sheet. And then we also process the serial sections with common histochemical stains, such as crystal violet for meso stain, Prussian blue for iron deposits, Luxil fast blue for the myelin, and Bielczewski silver stain for axons and neurofibrillary rectangles. There is a subset of 8 tissue blocks that were scanned prior to the histology processing with a 70 animal scanner using 6 sequences at a very high resolution. The Prussian blue staining was registered to the T2 turbo-rare MRI using the manual landmarks, and this was followed by an automatic finding error registration to the parallel MRI that was caught using the vibratom. And then we assessed 2 histology variables, such as the ease of manipulation of the tissue using 3 signs. So if the sections were teared, rolled up, or very sticky. And we scored the samples from very difficult to very easy to manipulate. And for all the antigens of interest, we scored the cellular distribution by looking through all the available labeled sections to assess the antigenicity preservation. And we found either no antigenicity, isolated cells, cells in light patches, or a homogeneous distribution across the sections. And we compared the observed frequencies of the 3 groups using chi-square. And we found that the salt-saturated solution showed more often brains that were difficult to manipulate. And we also found that the alcohol formaldehyde fixed brains were more often easy to manipulate, but there was no statistical difference between the three groups. For the cellular distribution, we found that the salt-saturated fixed brains showed a significant higher occurrence of isolated cell distribution for neuron, and more cases with no antigenicity at all for the IDA1 and the PLP. For the NBF fixed brains, we showed significantly more cases with an absence of neurons and astrocytes, but this did not retain significance after the Bonferroni correction. And finally, the AFS fixed brains showed mostly more cases with cell patches or even homogeneous distribution for the 4 antigens, as we can see in the 4 panels, reflecting a better antigenicity preservation in general. Overall, the cells were well preserved with the crystal violet and the Prussian blue staining, and the myelin fibers were also visible in Luxil fast blue staining for the 3 specimens. The Bielczewski staining labeled the neurofibrils and axons in brains that were fixed with the 3 solutions, so everything was fine. However, we found that the salt-saturated solution provided brains with worse cellular contrast and smaller cells, as you can see in the Nissl stains here. The MRIs show very good gray to white matter contrast in all the specimens, as you can see in the figure here, which reflected a sufficient MRI quality in brains fixed with the 3 solutions. So MRIs are feasible with these kind of brains. And the registration process was also good in all specimens. However, the SSS fixed brains that are here were more difficult to register, but still feasible and of good quality. I need to mention that. This was probably due to their poorer cellular contrast that we saw in the histochemical stainings, and also the ease of manipulation that I showed you was more difficult, and that led to tissue tears, which could enhance a more difficult registration afterwards. And we used salt fixed blocks that was cut close to the frontal horn of the lateral ventricle, and that showed periventricular white matter hyperintensities on the full brain image that was acquired prior to this project. We wanted to colocalize the dark spots of the T2 star map, and the blue spots found on the Prussian blue staining, in which both modalities could show iron deposits. So our goal is to eventually quantify the number of blue spots on Prussian blue stainings automatically, and then correlate them with the qMRI maps. Now that we have figured out the registration with multiple staining, we will also assess the relationship between different qMRI measurements and histology information using qMRI-AB, which is the open source tool for analyzing qMRI data in order to extract T1 and T2 relaxation maps, such as the mono T2 quantitative map or the myelin water fraction, and compare obviously with our co-registered histology image. Here we chose Biochelski to show the axonal sheet assessment, but we could also use the Luxel fast blue for myelin sheet. So in conclusion, histology, MRI, and registration between these two modalities are feasible and of good quality in brains that were fixed with two solutions used in gross anatomy labs and one typically used in brain banks. We have also established protocols on human brains that were fixed with different solutions, which could increase the amount of brain tissue available to neuroscientists who could use brains from gross anatomy labs instead of specimens from brain banks. And in addition, our MRI to histology registration pipeline might be a good tool for neuroscience research and a potential gold standard diagnosis. So I would like to thank you for your attention and the organizing committee for this presentation opportunity. I would like to thank my co-authors who did an incredible job, especially with the registration pipelines, and the incredibly generous donators from the body donation program of our anatomy department, our collaborators from the Douglas Bell Brain Bank, and our funding resources. So if we have a little bit more time, I can answer your questions. Thank you.
Very impressive work. Thank you so much for your presentation. Do we have any question, Mark?
Not at the moment, but personally, I'm curious to what extent you and your lab are planning on making the tools and pipeline, everything you've developed open source, and what you guys are thinking about that topic.
Yeah, I think the open source tool is the main goal. What we want to do is eventually to go backwards to the in vivo scan. So the tissue that we obtain in the anatomy lab, we sometimes have in vivo scans, and our goal is to then scan the head in situ, then extract the brain and scan it again ex situ. And then we cut the blocks, we scan them with the 70 MRI, as I showed you, and then we will register the histology to the block to the ex situ, and the ex situ to the in situ. So all of this registration pipeline might be a very nice tool for going back and co-registering the histology to the in vivo eventually. So yeah, it's obviously the goal to have the open source thing for everything of the registration pipeline.
Yeah, that sounds great. I think it would definitely be beneficial to a lot of people, especially in our lab. One question here from Dr. Hagberg. She asks, how about cell morphology? Was there a difference between solutions?
Yeah, we did a lot of work looking at the more of the shrinkage of the brain. So in different part of the brains, we acquired pictures, and we looked at the volumes of the cells, mostly the neurons for now. Yes, there was some difference between the solutions. So obviously we have a difference of shrinkage between the different fixatives. This is a work that will that's in progress actually, and that we will have a master student working on furthermore. And yeah, the cell morphology, we also did some immunofluorescence with the astrocytes, and we found that the processes of the glial cells were not always as long or as short, but we didn't have any quantitative results for now, but it's obviously very interesting. Yeah.
Okay, thank you. I think we have time for one more quick one. Dr. Nair asks, what is the pipeline you use for MRI histology registration? Is it automated or manual registration?
We started with a manual landmarks registration, and then there's a fine automatic registration too. This is not my part of expertise. I'm more like the histology part, but yeah, it's what they are doing for the registration of everything. If it's the block or the heads, whatever, they always use the manual and the automatic at the same time.
Okay, thank you. Very cool work.
Thank you.
Great. I just have a question quickly, by curiosity, like what is the timeline between the death of the participant and after like all the processing that you do and register?
Okay. Yeah, okay, so you mean the postmortem interval, like between that and between the fixation? Yeah. Okay, so we actually measured the postmortem interval between our brain bank specimens and our anatomy lab specimens. The brain banks were significantly longer than our specimens in anatomy labs. Our PMI mostly goes from maximum 48 hours of fixation. Yeah, we mostly have between 0 to 24 hours. Usually the fixation is done, but it depends on where they come from. Yeah.
Great, thank you.